Hi guys, good evening. I am Ilangovan, onlineaxi.com personal for teaching zoology. So we started the new concept of evolution in the last class. We talked about the various theories regarding the origin of life. But the most accepted theory of origin of life, you know that one, the chemical origin of life. That was first initiated by Parry and Haldane, and that also experimentally proved by two scientists, one from America, another one just actually from England. Harold Urey and then Stanley Miller experimentally proved the chemical origin of life as proposed by Haldane and Alexander Parry. So Miller in the laboratory created situation, similar conditions like that of the primitive atmosphere. So he created electric spark, the electric discharge, the one which provides energy. So in the past we had energy in the form of lightings or in the form of UV radiations or in the form of radiations, which are responsible for actually the union of different elements to form the complex molecules in the past. The same way we want to provide energy for the union of different actually the components. He provided electric discharge in a closed flask. See that one here, it is the electrical discharge, that is the one which provided energy for the union of various molecules. So it just actually sent the molecules like ammonia, just hydrocarbon like methane, hydrogen gas, and also water vapor. So this is the experimental setup. Water vapor is provided by actually the boiling of water in a conical flask, and that is being normally sent to or towards actually there is an electrical spark area, and also the ammonia, then methane also supplied, and all these components combined together. So by unifying or by uniting all these molecules with the help of energy provided in the electric spark, so Miller was able to isolate certain primitive and simple amino acids like glycine and alanine. So with the experimental proof, now the chemical origin of life has been completely accepted by most of the scientists. So anyway, we have formed the life just by means of abiogenesis, nothing but the formation of organic molecules from inorganic constituents. Then the diversification of organisms occur by phenomenon of biogenesis. So anyway, we have the life could have originated from the non-living organic molecules, and which were preceded by, you know that one, the chemical origin, nothing but the diverse formation of different organic molecules by means of actually active forces, the external forces, like electrical forces, that is the lightings or UV radiations, and other actual physical forces responsible for the union of such molecules to form the first life. So that is called what we can say the chemical origin of life, or simply we can say chemical evolution. Then, so followed by actually the chemical evolution, once the simplest molecules that what we have, namely the macromolecules like carbohydrates, proteins, amino acids, the nucleic acids were formed. Then we have the formation of the living organisms, the first living cell, what we call that is the protocell. And that process of formation of the first living cell is called biological evolution. And what are the steps normally we have during the process of biological evolution? One, the formation of prebiotic structures, the living structures. And these are all called the protobionts. The prebiotic structures, these are all called the protobionts. These are all nothing but the first formed living molecules with limited metabolic activity. They are not having much actually complexity, simple structures, but they have some ability normally to form certain chemical components to lead their life. Then, suppose you want to have the biological evolution, or for the evolution of any living thing, or for the origin of any new life, we need three components. One, the free genes, or we can say the replicators. These are all nothing but the self-reproducing structures, or self-multiplying structures, or self-multiplying molecules. So they are simply called as the free genes or the replicators just in the beginning. And these replicators normally underwent some sort of actually modifications. That is nothing but some sort of changes in these molecules, namely the free genes, and that is what we can say the error. So while actually the multiplication occurs, we have the copying or multiplication of genes also, the DNA also, or the genetic materials. So while copying, what will happen? Copying here is nothing but actually the replication process or the duplication process. A single molecule has been converted into two molecules, or the duplication we can say. And during that process, errors may occur. So that is a must. So errors may lead to a change in the structure of the genetic material. Ultimately, we have the formation of new species. And that error simply called as a mutations. So the second event, so first one, we have the raw material, namely the free genes or the replicators. While these molecules are undergoing replication process while copying themselves, some sort of error must be needed. Then only we can get a new genetic material. And that error is nothing but mutation. The error is nothing but mutation. And thirdly, we need a continuous supply of energy for the formation of such molecules. And all such molecules are being formed only in water. So they should be isolated partially from the environment. Then only we can get the aggregates to form a life. So the three components for any biological evolution, the replicators, or we can say the free genes, then some sort of changes, what we call this one as errors, nothing but the mutation during the course of time in the genetic material caused by various factors like temperature. And also we need a continuous supply of energy for the formation of molecules. And once the molecules or the aggregates formed in the marine water environment, they should be isolated from that environment by partial method. Now, how is mutation normally caused? So in the past, mutation occurred because of high temperature. So we have seen already high temperature prevalent in the atmosphere, the primitive atmosphere. That high temperature was responsible for causing mutation in the molecules, leading to the formation of new molecules of different types. So by mutation, due to temperature, many different molecules of different combinations have been produced with the different properties. And the next one, what I mentioned already, the partial isolation. So this partial isolation had been attained within the aggregates. So within the aggregates, actually the aggregates are nothing but subprotoplast like substance aggregated. And these aggregates, actually the partial isolation happened within these aggregates of artificially produced protobiotic molecules or prebiotic substances or protobions. So partial isolation normally attained with these aggregates, and these aggregates have been given different names. The aggregates, nothing but actually the organic molecules somehow dissolved in water, forming a semi-fluid or semi-solid substance. And there are two different types of aggregates as given by two different scientists. So these aggregates are nothing but protobions, commonly called, or prebiotic substances. The protobions may be called as co-aggregates by a pairing. Co-aggregates, the aggregates that is in the marine water. So it was also called as microsphere by Sidney Fox. So the co-aggregates and microspheres, nothing but the aggregates, they are also called as protobions, the first formed living substance in the marine water. And they have been isolated from, that is actually the water, get accumulated to form a complex semi-solid material, the first living substance. Now what are microspheres? When compared to the co-aggregates, the microspheres are also aggregates, but they are all provided with some membrane around them. But in the case of co-aggregates, there are no membranes, but the microspheres are provided with some membranes, so that's why they are called membrane-bound structures. And they have the ability to undergo multiplication. How? By means of budding. So by means of budding, they gave rise to more and more number of microspheres. Ultimately, then we have received the first result, except. So all the life forms, actually, all the life forms normally we have originated only from the marine water. All life forms only just from marine water, that too in water. So life originated only from water, not from terrestrial actually matter or terrestrial thing or terrestrial actually area. So don't forget, life originated only from water, and that too from marine water. All mixing occurs only in the marine water to form a hard dilute soup or prebiotic soup. And these were the components formed because the various components, the macromolecules dissolve in marine water, and also the accumulation of various elements found in marine water. Then only we got the aggregates in the form of microspheres or just co-aggregates. Now, so from the actually protobions, the microspheres and co-aggregates, Now we have the formation of the first living cell. First living cell. So what happened during the course of evolution? So I have given here in the form of sequence what happened during the course of formation of a new living thing. So first you know that when the formation of giant molecules, the macromolecules, the biological macromolecules in the form of RNA, proteins, polysaccharides, nucleic acids, generally we can say various other hydrocarbons. So the giant molecules were first formed in the marine water in the form of RNA or DNA or proteins or polysaccharides. And from these structures we have the formation of the free genes. And the free genes get surrounded by means of membrane to form the first protocell or the first living cell, what we call this one anaerobic heterotroph. So the first formed living cell, the protocell, is nothing but anaerobic heterotroph. So it utilized energy, it obtained energy by means of anaerobic method. That you didn't have a chlorophyll, that is why it's called a heterotroph. So the first formed protocell was nothing but anaerobic heterotroph that formed nearly 3 billion years ago. The first protocell. Life originated nearly more than, actually know that one 4 billion years ago, the first life. But the first protocell, the first living organism originated nearly 3 billion years ago. And the anaerobic heterotrophs later soon transformed into chemoautotrophs. They obtained energy by the oxidation of inorganic chemicals. So, and later on, by the formation of the bacterial chlorophyll from the porphyrin compounds from the marine water. We have iron porphyrin as well as magnesium porphyrin compounds. The magnesium porphyrin compounds present in the seawater then evolve into the first chlorophyll, what is called the primitive chlorophyll, what we had in the case of blue-green algae, simply called as the bacterial chlorophyll. So the chemoautotrophs, by just making or by just actually manufacturing the bacterial chlorophyll, a primitive chlorophyll from the porphyrin compounds from the marine water. So as a result of that one, now we have the evolution of the primitive photoautotrophs. Because of the evolution of the chlorophyll, the bacterial chlorophyll, and the first formed living organism now synthesized the food by using the light energy. And that is because of the blue-green algae. That is because of the what is called actually the bacterial chlorophyll. And that was developed in the case of blue-green algae, the cyanobacteria, what we call this one. Then later, the primitive chlorophyll, the bacterial chlorophyll, has been transformed into the higher phototropic chlorophyll complement. There is nothing but the true chlorophyll. So the true chlorophyll made their origin from this normally the primitive, actually the phototropic bacterial chlorophyll, and that had been transformed into just actually the true chlorophyll. As a result of this one, the evolution of higher photoautotrophs resulted. So primitive photoautotrophs transformed into higher photoautotrophs by the evolution of the true chlorophyll. So now the photosynthesis process had been started by these living organisms because they had the chlorophyll. So that what would happen? The release of oxygen occurred because of photosynthesis. Now the oxygen gradually released into the atmosphere. The free oxygen now oxidizes. That is the methane into carbon dioxide. So we have received also carbon dioxide. As a result of accumulation of oxygen, what we have developed, that is a formation of ozone layer over the atmosphere, and that resulted in preventing actually the intrusion of the ultraviolet rays to reach the troposphere. You know that when the ozone is acting as a screen, preventing the entry of that is ultraviolet radiations to reach the troposphere. So at present you know that when the atmosphere is called oxidizing atmosphere because we have high amount of oxygen in it. So formerly the atmosphere, the primitive atmosphere was considered as a reducing atmosphere with a high concentration of hydrogen. Now because of the oxidizing atmosphere, what would happen? There is no further evolution happens now. So there is no process of new life origin at present. The reason for that one, the atmosphere is called the oxidizing atmosphere, having the property of oxidation As soon as any new molecule being formed, it will be oxidized. So in the past, we had reducing atmosphere, there was no oxygen, there was no free oxygen. That is why once a new life came to this earth, that was not oxidized, that was persisting and leading to the formation of further actually succession of life. So now the atmosphere is called oxidizing atmosphere, and there is no evolution of life at present. This is because of the oxidizing atmosphere having the property of oxidizing any new material or any new life product formed in this world. So far you have seen actually what are the various concepts regarding the origin of life. So we have formed the origin of life by means of abiogenesis and also the diversity of living organisms by a process of biogenesis. That is nothing but formation of life from pre-existing organisms. Now we have to provide some support in favoring the organic evolution. So various evidences have been made by different people in different fields to just expose. Actually, that is a favorism shown by the evidences in support of organic evolution. Now it was Darwin who has given more importance for the geographical distribution of animals. So that is why he documented zoogeographical actual distribution of animals as an evidence for evolution. So not only the distribution of animals, but also he took the fossils, the fossil records as evidences in support of evolution. So he took the distribution of animals in different, not only the fauna but also flora, and also it took the fossil records as evidences of evolution. So in addition to these two evidences, either by means of geographical distribution or by means of what is called the fossil records, there are also evidences in support of organic evolution from the fields of comparative anatomy, morphology and comparative anatomy, embryology, we have physiology, biochemistry, biography, and also we have taxonomy, the classification, also support in favor of evolution. Now I will tell you more about what are the different fields of actually evolutionary, just fields of biology, which provided evidences in support of organic evolution. So what is the meaning of support? That means all organisms have been descended from a common ancestor. That is a concept. So there is a common ancestry for the present day organisms, and that was supported by a number of evidences from different fields of biology. That is anatomy, then morphology, embryology, physiology, even biochemistry, biography, and also just about the classification, nothing but the taxonomy. Now I will take what is the role of morphology and comparative anatomy in support of organic evolution. So we are tracing evolution relationships among the organisms. What is the meaning for that one? I mentioned already. All present day organisms have been descended from a common ancestor. So there is a common ancestor. So that is why we are finding or tracing some evolution relationships between different groups of organisms, though they are different in their morphology, in their attitude, activities, etc. Now first let us take. So what are the evidences from comparative anatomy? That is comparative morphology and anatomy. You know the meaning for morphology. So nothing but actually the external appearance. And anatomy, nothing but the internal and external organization. So we are calling this external organization as morphology, internal organization as anatomy. So the studies of external and internal organization of animals and comparative anatomy of different actually animals, they provide evidences in favor of evolution or organic evolution. External and internal organization referring to the anatomy and morphology, and we are comparing the anatomy, comparing the morphology, how far they are relating to each other, then exhibiting the common ancestry. Now while we are studying about this comparative anatomy and morphology, what they revealed us. So they normally reveal that. So when you are going for evolution, the anatomical characters become more and more complex progressively from lower to higher group of organisms. So starting from amoeba up to man, you see there is a progressive change in the anatomical structures. So from simple to complexity occurs while we are talking about the anatomical evidences and morphological evidences. So anyway, the anatomical characters become more and more complex, slowly and progressively from lower to higher group of organisms. Now again, suppose you take the characters, the morphological characters or anatomical characters inherited with animals. So the animals inherited the anatomical similarities. How? Because they had a common ancestor. So the animals inherited their morphological and then anatomical similarities from a common ancestor. That is why we have, though we are all different from one another, different species, they are all actually looking alike in one aspect because they have been descended from the common ancestor. So animals inherited the anatomical similarities only from a common ancestor. So these are all the two facts what we understood from the facts revealed by anatomy and morphology. So under this comparative morphology and anatomy, we have to talk about certain organs, homologous organs, analogous organs, vestigial organs, etc. Now what do you mean by homologous organs? Suppose we are taking the limbs of a horse, the forelimb of man, the forelimb of whale, or even in the case of bat, or in the case of birds. So in all vertebrates, if we are taking actually the limb skeleton, the limb skeleton normally has the same basic fundamental structure. So an example of the forelimb, we have upper limb humerus, then the forelimb normally just actually the forearm consists of outer radius and inner ulna. We have the wrist having the corpus, then the palm region, the metacarpus, and then the digits being formed of a number of phalanges. This is a basic pattern, basic structural arrangement. But you see, though they have the basic structural arrangement, the similarity is there with reference to the structural arrangement of the organs and also the origin. And there are different organisms. Say an example, the horse or whale or the bat or birds. They are using the forelimbs for different purposes. So, all the bones have been descended from what is called a mesoderm. Though they have been descended from a mesoderm, though they have the same basic fundamental structure, if you are taking the function of a horse limb, it is used for running, and for whale or dolphins, it is used for swimming, and for birds and bats, it is normally used for flight. So, the organs which are having similar in origin and are built on the same fundamental pattern but perform different functions are called as the homologous organs. Similar origin, similar basic structural pattern, but different functions. Similar function, similar fundamental structure, but different functions. Such organs are called homologous organs. And normally the homologous organs explains one phenomenon, what we call this one divergent evolution or evolutionary divergence. We'll come to this later. That means from one type of organ. So different organs actually one type of fundamental structure. Different structural changes happen, resulting in the formation of different structural organization for doing different functions. So anyway, from one central point, many deviations occur. This is what we can say the divergent evolution. Formation of different species from a single species. That is also called as divergent evolution or evolutionary divergence. So, the same fundamental structure, but it has been modified to form different external organization to perform different functions. So, origin-wise same, then structural-wise same, but functional-wise different. So, that is why it's called as divergent evolution. From a single stock, many different organisms have been diversified, and this is what we can say evolutionary divergence or divergent evolution. So homologous organisms, homologous organs exhibit the phenomenon of divergent evolution. So I mentioned already the main example in all the vertebrates, mammals, birds, reptiles, and amphibians. They have four limbs, upper limbs and then lower limbs. If we are taking the basic structure of the limbs, either the forelimbs or hind limbs, it is similar. Though the limbs have been modified in different vertebrates to perform various functions. So this is what is happening in the case of all these four limbs. So the basic fundamental structure of the forelimb, or just if we have the hind limb, same. Having that one upper arm, forearm, and then hand. In that one we have the corpus, metacarpus, and phalanges. But they are modified here and there in such a way that they are doing or they are performing different functions. So the basic structure is the same in the case of all the vertebrates regarding the limb skeleton. but variously modified to perform different functions. Now, what are the other examples? So, one example, the limb skeleton in the case of vertebrates, including the mammals, birds, reptiles, and amphibians. Now, what are the other examples? In the case of invertebrates, if you are taking cockroach or any other insect, so the leg of insect, the cockroach, or we can have, for example, the grasshopper, or we can have, for example, the praying mantis, beetles, the butterflies, the moths. So in all cases, they have legs. The legs are five-jointed: coxa, femur, trochanter, tibia, and tarsus. But the legs are variously modified. For example, the case of one insect, water diver, the one which is swimming on water surface or diving into the water, the legs are actually modified just like a oar. And in the case of the water spider, an insect floating on the water surface, the legs are actually very thin for floating to reduce the weight of the body. So the legs are variously modified to perform different functions in different kinds of insects, but all are having the same basic fundamental structure with the same basic plan. Then another example, the mouth parts of insect. So if you're taking the mouth parts of insect, any insect, it is being formed of upper what is called the upper lip, the labrum, upper lip, labrum, lower lip, labium. Then we have some masticated structures, what we call this one, the mandibles and maxillae. So these are all the fundamental structures like labrum, labium, then we have maxillae and mandibles. And in all insects, we have the same basic fundamental structure. But from the primitive actually mouth parts, we have diversified different groups of mouth parts or different types of mouth parts. For example, in the case of housefly, it is called sponging and sucking type of mouth parts. In the case of a butterfly, it is called siphoning type. In the case of cockroach, it is of biting and chewing type, siphoning type, biting and chewing type. And in the case of mosquitoes, it is called piercing and sucking type. The same structures, the same basic fundamental structures of mandibles, maxilla, labium, and labrum. But these structures are variously modified to perform the function of sucking or piercing or sponging or even siphoning, etc. So the basic structure is the same but modified to perform different functions. So it is also an example for that is homologous organs and the phenomenon homology. And normally such homologous structures help to normally identify an evolution relationship between apparently different ancestors. So they have different ancestors, but they are all just actually coming back to perform one function. Even we can say that is they're converging at one point or deviating from one point. So anyway, any homologous structure normally helps to identify an evolution relationship between apparently different ancestors, not the same ancestor, apparently different ancestors. That is very important with respect to the homologous structures. Now here given the mouth parts. Now the primitive type of mouth parts, the basic one, the ancestral mouth parts is nothing but the biting and chewing type. See, in the case of mouth parts, we have the labrum upper lip, labium lower lip, the mandibles for cutting and chewing, then we have the maxilla. So these are the basic components of mouth parts of any insect. So in the case of cockroach, we have just actually the primitive type of mouth parts, what we call this one, the biting and chewing type, even the case of grasshopper also. Now it has been actually diversified into either sponging and sucking type of mouth parts in the case of housefly, or actually it has been modified to siphoning type, as in the case of moth and butterfly for sucking the honey from the flowers, or it is being converted into or transformed into piercing and sucking type in the case of mosquitoes for injecting its needle-like structure, what we call this one proboscis, and sucking the blood. This is called piercing and sucking type. So the basic structure is the same. In all cases, we have the same fundamental structures, but they are modified according to the functions. And that phenomenon is called homology. So the mouth parts of insects represent homology. Okay. Now here given the colorful structures, what I represented in the form of previous one. And now the sponging mouth parts then the siphoning mouth parts in the case of that is butterfly, and sucking mouth parts even in the case of bedbug also you can have sucking type of mouth parts, injecting its needle-like structure and sucking that one, and chewing type of mouth parts. What is given in the figure is nothing but an insect, praying mantis, praying mantis. So that is nothing but an insect similar to cockroach in its resemblances. It is called as a praying mantis. So these are all the different photographs showing how far actually the homology occurs in different groups of organisms. So I mentioned already homologous organs explain the phenomenon of evolutionary divergence or divergent evolution. It also exhibits another phenomenon, what we call this one adaptive radiation. Adaptive radiation. So what do you mean by adaptive radiations? Normally the homologous organs normally explain the phenomenon of adaptive radiations, that is from a common ancestor. So from a common ancestor, what we had in the case of mouth parts, you see that one, the biting and chewing type of mouth parts, which have been diversified to form different types of mouth parts and different species. So that phenomenon is called also adaptive radiation. So what do you mean by adaptive radiation? So there are certain evolutionary processes are taking place. So that evolutionary process which gives rise to a new species, and a new species was given rise to by means of what is called the evolutionary process. That new species normally adapted to various habitats and also adapted to various ways of life. That phenomenon is called adaptive radiation. A phenomenon, an evolutionary process which gives rise to a new species. That species is normally adapted to various habitats, either environmental habitats of water or land or even just the trees, like that. So they are adapted to different habitats. So we have a process of evolution which is operating, and that will give rise to a new species. That one is adapted to different habitats, and also the ways of life or mode of life is called adaptive radiation. Or we can say another way, the process of evolution of different species in a particular geographical area, just normally starting from a point. There is a central point we have. This is the ancestral form, and from this one we have the diversification of different groups. This is what is called adaptive radiation. One central point, the starting point. So normally from that point, literally radiating different groups of organisms adapted to various habitats or geography. That phenomenon is called adaptive radiation. The evolution of different species actually by a process of evolution that is happening in a given geographical area, starting from a point, a central point. It is nothing but an ancestor. From the ancestral form, many new forms have been originated. So we have so many examples for this adaptive radiation. So it was reported by Charles Darwin in his findings in one island in Pacific Ocean, what is called Galapagos Islands, and that island is called as living laboratory of evolution. Galapagos Island, that is called living laboratory of evolution. So the first example for adaptive radiation, Darwin's finches, a kind of sparrow. So he observed more than 20 different species of that one finches, the sparrow, about Geospiza. The name of the species, genus name, Geospiza. So they are called as Darwin's finches, nothing but a sparrow. So he observed this Geospiza, more than 20 different species of Geospiza in island, what we call this one Galapagos Islands in Pacific Ocean, while he was going for a voyage in search of taking some statistical data in HMS Beagle from 1831 to 1836 for nearly 5 years. So he visited this one, that island, Galapagos Islands. Till today we have a number of evidences in support of evolution from that one. That is why that island is called living laboratory of evolution. Living laboratory of evolution. Then another example, we have the Australian marsupials, the pouched mammals. See that one, kangaroo, the papazians, then koala, that is what is called the teddy bear. Then also we have, for example, the Tasmanian wolf. So many examples. Australian marsupials. And also the Australian placental mammals, the highly evolved mammals, without a pouch. So the first one pouched mammals, the second one the placental mammals of advanced type belonging to the placentalia, or you see that one the eutherians, the true mammals. So all these actually evidences are in favor of adaptive radiation. Let's see the pictures of this one. Now the homologous organs I mentioned earlier. See that one, I am taking the four-limb skeleton humans. Then we have the cat, then the whale and the bat. In all cases, you see that one, they have upper limb skeleton or upper arm bone. That is what is called the humerus. Then we have just actually here it is given nothing but actually what is given, the hind limb skeleton is given regarding this one. So we have, let us take this femur, tibia, or just like that. And if you are taking the four-limb skeleton, that is humerus, then radius, ulna, then we have the carpals, and then the metacarpals, and we have the digits in the form of phalanges. See that what in all cases, see the color. See, this is what we have, outer radius, and now this is the ulna, the yellow colored one. So the different colors normally indicating the similarity in structure of bones. So in all cases we have the basic structure, but they are modified. What for? For doing different functions. In the case of human, for multiple walk. In the case of cat, it is used for also just walking. And in the case of whale, it is called as a flipper. It is called as a flipper. It is used for swimming. And in the case of bat, you know that when the wing is called as patagium, the skin fold, and with the help of which it can fly. So one for flight, one for swimming, one for walking, and one for multiple walk. But all have the same basic fundamental structure. The functions are different, but the structural arrangements same, origin-wise same, and such organs are called homologous organs. Another figure showing the arrangement of different roles in some more animals. See the first one, human, upper arm bone humerus. So it is given, you see that one, humerus, radius, ulna, the carpals, metacarpals, and phalanges. So human, it is used for multiple purpose. A lizard, it is for, you know, that one, crawling. Cat, it is used for, you know, that one, walking. And then whale, it is used for swimming purpose. Bat for flight, and frog for hopping on land, and bird also for flight. So the same basic structure with the slight modifications. For example, if you are comparing the four-limb skeleton of human with that of bird, so we have 5 digits. But in the case of birds, you see that one, the number of digits only 3. So 3 digits only present in the case of actually the four-limb, which is modified into, you know, that one, the wings. That is a slight modification only because of the adaptation for flight. But in all cases, they have the same basic fundamental structure. That is why they are considered as the homologous organs. Origin same, then function different. Now here what I mentioned, the adaptive radiation. So the process which gives rise to different species which are adapted to the various habitats and also the ways of life. That is why they have diversified from a central stock. Now here given the marsupials, the Australian marsupials, there is a central stock. So from that central stock we have the radiation. That is nothing but many species have been diversified for their mode of life, for the different habitats. You have the Tasmanian wolf, the tiger cat, the just what is called the banded anteater, the marsupial rat, kangaroo, the famous one, you know that one, the pumbet, the bandicoot, the koala, what is called normally that is Australian teddy bear, then marsupial mode and sugar glider. So these are all the different types of marsupials, but they have originated from one common ancestor. So they have been diversified to adapt themselves to different habitats and also the way of life. So this is an example for adaptive radiation. Now this is Darwin's finches. See, the primitive ancestral form was nothing but a seed eater. So from the seed eater variety, we have developed a number of species. Some of them are eating on buds and fruits. Some of them are eating on grubs, nothing but the insect larvae and other simple organisms. And some of them are using the tool. You see that for making use of the tool, for just actually making or constructing the nest, or for just looking after the young ones, or even for gathering the food. And some others are feeding on insect, and still some others are feeding on leaves. So according to the mode of feeding, what will happen, the beak has been modified. From a simple nut eater, or sea eater. See that one, the beak has been modified. You could see in this picture the different types of beaks. And these beaks are formed for doing different functions. According to the mode of feeding, the beaks have been modified. But the same structure, upper and lower, made up of horny only, horny material. The origin-wise same, the function is only different. The structure also different. Somewhat basic pattern, but they are somewhat different. So that is about the Darwin's finches, exhibiting homology as well as adaptive radiation. Here given the colorful picture of many species. So from the ancestral nut eater or seed eater, we have developed mainly insect eaters, then we have mainly seed eaters, and also the buds and the fruits eater, and also feeding on cactus and seeds. So in all cases the modification is with reference to only the beak first. The second modification also took place in the nature of the color, because in the case of Galapagos Islands the background is not uniform. The background is different. So according to the nature of the background, they have modified their feather colors also. So from a single species, here we are talking about only there is a beak, but also if you are taking the feather color, the feather color has been modified according to the nature of the background. You could see the color of the different heads of these birds. So anyway, Darwin's finches in Galapagos Islands, an example for adaptive radiation. So I mentioned the placental mammals as well as the marsupials found just in Australia, and both exhibit adaptive radiation on their own way. But some of the marsupials and some of the placental mammals are unique or a little similar, because they are all converging towards one point, towards one adaptation, though they are exhibiting divergent evolution, adaptive radiation. And these two different groups of organisms are converging towards one point for leading their mode of life. So we have different marsupials from a single ancestral form. We have different placental mammals from a single placental ancestral form. But both of them are moving towards one point. They are converging towards one point because of actually the mode of living. So that is why here I have given the statement. Some placental mammals also exhibit adaptive radiation, and each of which appears to be similar to a corresponding marsupial. So one marsupial, for example, if you are taking the Tasmanian wolf, it is a marsupial. It is having a pouch, you know that one, the marsupium, pouched mammal having a pouch. But the placental mammal doesn't have that pouch. But both are converging towards one point. For leading a life, both are diversified from a common ancestor. If you are taking one, for example, placental mammal, it is diversified following adaptive radiation. And similarly, if you are taking the marsupials, they are also diversified from a common ancestor. But both the placental wolf and the Tasmanian wolf are almost similar in their external appearance. The name only different, and also their organization. And both are converging towards one point and exhibiting what is called, we can say, the convergent evolution. Each one is representing adaptive radiation and divergent evolution, but both of them are moving towards one point for leading a common life, just based on the nature of the environmental habitats, the mode of feeding, etc. So they exhibit what is called the convergent evolution. So both are distantly related species. So the evolution of most distantly related species is called convergent evolution. They are converging towards one point for leading one common life, that's all. But they have been descended from two different ancestors, but they are converging towards one point. That is why here the statement is like this. So the placental mammals also exhibit adaptive radiation, and each of which appears to be similar to a corresponding what is called Australian marsupial. Here is one example. The placental wolf is similar to that of Tasmanian wolf, but both are different. Each individual is exhibiting adaptive radiation, but together they represent what is called the convergent evolution. So I will show the pictures here. Now we see that one. The left side we have the placental mammals, on the right side we have the Australian marsupials. Both are actually living in Australia. And you see that one, this is the one exhibiting adaptive radiation. The placental mammals from the common ancestor. Different types of placental mammals originated. You see the mole, anteater, the mouse, the lemurs, the flying squirrel, then just also cucumbat, and also wolf. They have been descended. They exhibited adaptive radiations individually. And also they are exhibiting convergent evolution together with Australian marsupials. Now the mole, the placental mammal, is more or less similar to the marsupial mole. It's also called as a mole. They are leading more or less common life, but different. Different species are different actually organizations. The anteater, placental mammal, this is limbed, another anteater. See, they are called anteater. They are eating the ants, but structurally, you see, the organization is different. Then the mouse, and then marsupial mouse. And then we have the lemur, and then just spotted, the animal, cuscus. And then flying squirrel, and this is flying phalanger. And this is bobcat, and then this one, the Tasmanian cat. And last one, I am taking the wolf. That is the one, you see that one. This is Tasmanian wolf, a kind of marsupial. And this is a normal wolf, what we know that one. That is a placental mammal. And both are different species, but both are converging towards one environment, namely the Australian forest, the Australian grasslands, and for leading a common life. And both are having more or less the same niche, the mode of habitat, the mode of feeding. See that one that is happening, wolf and Tasmanian wolf living together. And there is also one question regarding this one. So all these mammals are living together peacefully. Either the marsupials or the placental mammals are living together peacefully. How is it possible? So even the animals are also without any exploitation, without any just competition. The reason for the animals which are living together peacefully in Australia, the reason for that one is mainly lack of competition. Though the animals are actually moving towards one niche, feeding on the same type of food. But normally what is happening here, actually, that is the lack of competition is there in Australia. That is where they are living peacefully. There are no, you know that one, the carnivorous animals, the powerful carnivorous animals like tigers and lions. So, because of lack of competition, the animals are living peacefully in Australia. So, individually the placental mammals exhibit adaptive radiation, and also the Australian marsupials. Together they exhibit convergent evolution, just converging towards one point for just mainly leading a common life. Now, in contrast to the homologous organs, so in homologous organs, you know that one, same origin, more or less same built pattern of structures, but functions different. So, in opposite to that of homologous organs, we have the analogous organs, and those organs which have different in origin. So that one origin same, here origin different. So the organs normally that differ in their origin and in development. Regarding the origin development, they are different but perform similar functions. Such organs are called analogous organs. So two different groups of animals. Say an example of insect and a bird. Both are having wings. Both are entirely different. If you are taking the wing origin of insect, that is different. It is nothing but the expansion of the cuticle. In the case of bird, it is nothing but a feathery covering. It is entirely different. It is made of bones, feathers and muscles derived from ectoderm and the mesoderm. So the origin-wise, the wing of insect, the wing of bird is different. But both are performing one common function, namely the flight. And such organs are called analogous organs. Organs which have different origin but perform same functions. So the origin different, development is different, but the function wise same. Such organs are called analogous organs. The best examples, we have the wings of birds and bats also. Though we have both are vertebrates, both are having, and for example, if you are comparing the four-limb skeleton of birds and bats, they are coming under the homologous structures because both are having the four-limb skeleton. But if you are taking the origin wise, actually the functional wise. So origin wise, the wing of birds and bats are more or less similar, having the bones. But if you are taking the structure and function, the structure. So in the case of birds, it is nothing but a skin fold. In the case of birds, it is nothing but actually a feathery arm. So we have to conclude birds and bats, the wings of birds and birds are actually nothing but analogous organs, not homologous organs. But it is very simple. Wings of birds and insects are analogous organs, both are entirely different. So, but in the case of wings of birds and bats, you know that the origin more or less same. But if you are taking the structure somewhat different, functions same. That is why we have to consider the wings of birds and bats analogous organs rather than homologous organs. Okay, so wings of birds and insect, then also the eye of octopus. So in the case of cephalopods, one group of mollusks, highly advanced mollusks, the eye is similar to the vertebrate eye, either in the case of octopus or in the case of sepia, other molluscan forms, sepia or loligo. So in all cases, these are all actually the molluscan animals, cephalopods, having eyes similar to that of vertebrate eye. That's a common character. But they are origin-wise different, functionally same. So meant for sight. So eye of octopus and mammals, origin-wise different, but both are used for sight. Then the flippers of penguins and dolphins. The penguins, you know that one, the flightless birds. The wings, actually, they are used for just swimming in water because it's a water body, you know that one, living in Arctic and Antarctic regions. But they're all called as the flippers. So the flippers of penguins, the just what is called the flightless bird, and the dolphins, the aquatic mammal, or we can say also the whale. These are also analogous organs. So the penguin, actually, the wings, the dolphins, and though they are all actually doing the common function, some of origin-wise different. So organs which have different origin, different development, but perform same functions are called analogous organs. Some more clarity for these things. So if you are comparing the wing, why the wing of bat and bird are considered as analogous organs, though they have the basic structural arrangement. So in structure, if you are taking the bat, so it contains actually the wing is nothing but a skin fold, and that is extending or stretched in between the two elongated digits. So the skin fold, not actually a muscular fold, it is a skin fold, it is not made up of muscles. We have the bones, but the skin fold, what we call so the patagium, is being stretched between the two elongated digits. It's not considered as a muscle fold, simply a skin fold. But if we are taking the wings of bird, it is nothing but a feathery form. It is nothing but a feathery form, or it is a feathery covering made up of muscles and the bones and also feathers found all along the arm. So, as a result of these two characters, if you are comparing these two, so if you are taking the designs of these two organs, the designs of the two wings, their structure, and also the components, thus making them different. So the design, the structure and the components, the design, the structure and components of these two are different. That is why they are considered as analogous organs rather than homologous organs. So I mentioned all they look similar. What is the reason? Because they perform one common function, namely that is what is called the flight, but their origins are not same. So, and this makes them actually analogous structures rather than what I mentioned earlier, the homologous structures. So in the case of bat, it is a skin fold. In the case of bird, it is nothing but actually a feathery covering made up of muscles, bones and feathers. There it is nothing but a skin fold with bones. And though they are all similar for doing common function, normally they are considered as analogous organs because their origins are different. Now here is the picture showing the analogous organs. So the first one, the bat wing. So let us compare first actually the wing of a bat and bird. So you see that one here we have the feathery structures with bones, and here we have the bones with the skin fold. So the skin fold originated from the ectoderm, and here the bones and muscles developed from the mesoderm. So made up of feathers, bones, muscles. Here made up of skin fold and bones, origin wise different, but structurally they are similar, but their origins are not common. That is why they are considered as analogous organs. Now we are comparing now the bat wing, the bird wing with what is called the insect wing. See, the insect wing is very transparent, you know that one. Its wing is being supported by many nervures, thick kind of structures which support the wings of insect. So I mentioned already the wings are nothing but the expansion of the cuticle. Nothing but the expansion of the cuticle. If you are comparing the wing of insect either with the bat or with the bird, they are entirely different from one another origin wise. But all the three are concerned with one function, what we can say, nothing but the flight. So different origin, common function. So here is a picture I mentioned just already, a colorful picture. Now this is, you know, that on the back, this is a sparrow, the hummingbird also we can see. Now this one, the insect, and this is the dinosaur, the primitive one which lived during the Mesozoic era, pterosaur. It also had wings, but never used for flight. It was used only for gliding. So we are comparing the wing of bat, wing of bird, wing of insect, and also one dinosaur, pterosaur, that lived in the past during the Mesozoic era. All were considered as, now also considered as, what are called the analogous organs. Now, what is the importance of such homology and analogy? I mentioned earlier, homology is concerned with divergent evolution, whereas analogy is based on convergent evolution, which is also called homoplasy. So, that is the main difference. So, homology or homologous organs represent the divergent evolution. Analogous organs or analogy represent what is called the convergent evolution. So, different species are converging towards one point for a common mode of life. And in the case of homology, the different forms of life, the different organisms have evolved from a single common ancestor. They are diversified to lead a different modes of life. So, what do you mean by actually convergent evolution? So, we can say simply the evolution of more distantly related species is called convergent evolution. For example, the insect and the bird are coming together towards one phenomenon, what is called the flight, but they are different. So anyway, the animals, if the animals are more distantly related, that evolution is called what is known as convergent evolution. So the evolution of more closely related species is called parallel evolution. For example, the placental mammals and marsupials exhibit parallel evolution. They exhibit parallel evolution, more or less closely related to some, but they are converging towards one point, leading a life, a common life. So anyway, if the animals are more distantly related, then the evolution is called, the process called convergent evolution. And the evolution of more closely related species is called parallel evolution. Then, I mentioned earlier, analogous organs are not anatomically similar. Anatomically similar. That is why, actually, though they perform different, so though they perform similar functions. So, analogous structures are formed as a result of convergent evolution. Analogous structures or organs are formed as a result of convergent evolution. So, different species are converging towards one point for a common mode of life. Here is an example of the wing of bat, as well as, for example, bird, and also, for example, insect. So, they are converging towards. So, analogous organs are resulted due to convergent evolution. Okay, that is one thing you have to remember. Then, vestigial organs. So, we have homologous organs, analogous organs, all are in support of what is called evolution. From comparative anatomy and morphology, we are talking about homologous organs, analogous organs, now vestigial organs. So, in our ancestors, we had certain organs which were functional, but at present, they are rudimentary, non-functional, reduced in age. And such reduced, rudimentary, non-functional organs, which were normally useful to the possessor in the past, are called as the vestigial organs. Suppose we are taking human beings, we have developed a number of vestigial organs. Our ancestors had a number of vestigial organs. For example, our ancestors had the ability to move the earlobes because of the auricular muscle. They also had actually the membrane covering for the eye, nictitating membrane, but it is being reduced in our case. Even the vermiform appendix, it is highly developed, normally in the case of our ancestors, for the digestion of cellulose. So those organs which were functional in the past, but now reduced, rudimentary and non-functional, are called vestigial organs. And normally these organs are highly developed in our ancestors, what I mentioned, and becomes non-functional in certain descendants. In certain descendants, they are non-functional. And if you are taking, for example, the cecum, one of the structure in our intestinal tract, it is normally reduced in our body, but it is well developed in the case of rabbit to digest cellulose. So our ancestors, which were eating on fruits and vegetables and other leaves, even just actually the vegetable materials, they had large cecum and also the tubular structure attached to it, what is called the appendix. And they were well developed because they were concerned with the cellulosic digestion. But at present, we are not using much just usage for digesting the cellulose, the cecum as well as the appendix much reduced. So anyway, the organs which were functional in the past but now rudimentary, non-functional, reduced are called vestigial organs. So according to one person by name Vida Sheep, a scientist, he observed more than 180 vestigial organs in human body. Some of them are, for example, vermiform appendix, the one which is attached to the cecum, highly reduced a tubular structure. If it is inflamed, you know that when we are getting the disorder also, appendicitis. And plicus semilunaris, towards the inner corner of the eye it can have. Just let me show the picture. See, here is a picture. So one example, the auricular muscles. So these are all the muscles placed in different places, anterior, posterior and superior. And these muscles were functional in our ancestors. That is where they had the ability to move the earlobe just like elephant. Now the second one, the vermiform appendix. So the vermiform appendix, you see that what is highly reduced, and also the cecum. And you see the tail region. The last part of the vertebral column highly reduced to form a fused structure, what is called the coccyx. And also the nictitating membrane. So towards the right side, we have the nictitating membrane in the case of apes. But here in our body, it is being reduced into a small reddish spot that is called plicus semilunaris. Even the wisdom teeth also highly reduced in our case. So these are all the structures concerned with what is called the vestigial organs. And as we have a number of vestigial organs in our body, man, we are described as moving museum of antiquities. As we have a number of just actually the structures, we are considered as a moving museum of antiquities. Then what are the other structures in different groups? Say in the case of wings in flightless birds, kiwi, ostriches, reduced eyes of deep sea animals and cave animals because there is no need for them to develop high powerful eyesight. And reduced pelvic girdle and hind limbs in the case of snake, python. And also we have some other structures. And there is one basic concept being used in the case of vestigial organ. It supports one concept proposed by Lamarck, use and disuse theory. So it is in support of use and disuse concept, namely actually proposed the third concept as proposed by Lamarck. So these are all some of the vestigial organs. We have also the case of python. That is a snake. So okay, just we see that one in the case of snake, the hind limbs and the pelvic girdle highly reduced. We have only the ischium bone and also the reduced femur. When you are comparing this one with that of our pelvic girdle and the femur bone, see the femur is highly reduced, and also see the pelvic girdle highly reduced. It is in the form elongated bone-like structures. So python is nothing but a snake, a longest snake. You know that one, it can consume even a small goat, consuming it as such without just it can swallow it and digest it for a month. So that is the longest one, you know that one, but not poisonous one, but consuming the whole body of an animal or any small animal like rabbit or the lamb of goat, etc. So anyway, we have the vestigial organ is here comparable to the pelvic girdle and the hind limb skeleton. The hind limb is reduced in the form of small white structure. Here it is represented, and the pelvic girdle, only the ischium bone alone present. It is also highly reduced, exhibiting what is called the vestigial organ in python, the snake. Because of non-usage, in the case of python, the hind limb is highly reduced, and so also that is the pelvic girdle. So with this I concluded some of the parts of actually evidences in support of organic evolution, mainly from comparative anatomy and morphology. So as time has been completed, I conclude, and you are ready to ask any question, or else you pose a question, we are ready to answer it. Okay, thank you. The class is completed. We'll meet once again in the next class.